Okay, so today's video is going to focus on active transport and then also answer the question as to why cells are small. If you recall from our previous video, we talked about passive transport introduced the concentration gradient and how naturally things want to move from a high to low concentration and then equal out into what's called equilibrium. Well, active transport is the exact opposite. Things move in a low to high concentration. So equilibrium is not happening because the higher concentration gets higher, the lower concentration gets lower. We call this moving against the gradient. And in order for this to happen, this requires energy. And our energy is in the form of what's called ATP. That's the energy molecule processed by the cell. Now we'll focus on two ways that this happens. One is using protein in what's called a protein pump, kind of similar to facilitated diffusion, but going from that low to high concentration. The second is what's called bulk transport, and this is when we are going to use vesicles. If you recall when we talked about the endomembrane system and how things get out of the cell, a vesicle fuses with the cell membrane and deposits those molecules outside the cell. Well, that's an example of active transport. It requires energy to do such. Now, ATP is our big energy molecule. As you can see right here, it is adenosine triphosphate, so 3 phosphates. And essentially, what wants to happen is we want to break that outer phosphate. It contains a lot of energy, break bonds, releases energy. Now with our ion pumps, once again, transport protein, so this is help to get things across. We're moving ions. Remember, ions are positive or negative charges. They cannot directly go across. So we have different ions that operate in an active transport setting. Basically, just remember sodium-potassium pump. That's probably our most well-known and most common example of active transport protein pump. What that does is that does play a key role in electrical signals among our cells. So when we look at the sodium-potassium pump, what you'll see in this picture is there is a higher concentration of Na plus outside the cell. There is a lower concentration of Na plus inside the cell. So we are going from low to high. So Na plus wants to move out of the cell. In contrast, when we look at the potassium ion, we have a lower concentration of potassium ions outside the cell and a higher concentration of potassium ions inside the cell. So as sodium ions are being moved out, potassium ions are being moved in. In order for this to happen, when we're going against the gradient, going from low to high, this requires energy to happen. So we would say that this goes up the gradient, or we would say it goes against the gradient. Now our bulk transport, we have two methods. We have what's called endocytosis. Endo simply means enter. Cyto means cell. So enter cell. This is where we're moving large particles into the cell. And we are using our vesicles to do so. So particles basically will come into the cell membrane. It'll form a little bit of a pocket. The cell membrane will pinch off, and we have a transport vesicle that'll go wherever it needs to go in the cell to deposit. Now you will have a lysosome that will use enzymes to break that vesicle down and further make sure that it is delivered to the correct location within the cell. On the other hand, when we talked about our endomembrane system, we talked about basically exocytosis, but we just didn't name it that. So exo means exit, cyto means cell, so we are leaving the cell. So when that transport vesicle is fusing with the cell membrane, it deposits those molecules outside of the cell. Once again, larger molecules, so it costs energy to simply make a vesicle and deposit them outside. So vesicle bulk transport. Bulk transport, if you are ordering something from UPS or FedEx, bulk transport costs more money. Same idea within the cell. So the question to be had is, which type of cytosis would increase the size of the cell membrane? Well, think about this. If you are entering the cell, part of that membrane is pinching off. So the cell membrane is getting smaller since it is pinching off. When you are leaving the cell, this is fusing with the cell membrane, vesicle, going out, exiting. So that means it is getting bigger. So it's kind of a give and take relationship because we have things coming in and out. So overall, it kind of evens out in the end. But exocytosis does increase the size of the cell membrane. So a little rehash. Passive versus active. Well, let's start with what we talked about today. The sodium-potassium pump is active, and then our vesicle transport, exocytosis and endocytosis, are active. So energy is required for all of your active. So everything else would be considered passive. So no energy required for passive. And remember, osmosis is dealing with the diffusion of water. And facilitated diffusion, once again, is using that protein help, which is what we also see with the sodium-potassium pump. So that essentially marks the end of cell transport. Now we're going to transition to cell size. Now cell size is focusing on the ratio of surface area to volume. When we look at sizes of things, prokaryotic bacteria is much smaller than a eukaryotic cell. So prokaryotic cell much smaller than the eukaryotic cell. Once again, kind of going back to origins when we talked about chloroplasts and mitochondria and kind of forming those eukaryotic cells. Chloroplasts and mitochondria are basically the size of bacteria. Kind of what we see right there. Kind of mimicking that, the endosymbiotic theory. Now, when we look at the problem with cell size, we have to look at those surface area to volume ratios. Tennis ball or soccer ball, which has a larger surface area to volume ratio? Well, this is going to be a tennis ball. There is more volume in a soccer ball compared to the surface area. The tennis ball itself has more surface compared to empty space in the middle, so we have a larger surface area volume. If you have a larger surface area, you'll probably have a larger ratio in comparison. So it comes down to, well, what limits cell size? Going back to this ratio. So we're going to look at in lab, specifically this type of investigation, this ratio, to show you, why cells are shaped very small. So what we want to look at is how much outside space there is, which is surface area, compared to space on the inside, which is volume. Too much space on the inside takes too much time for things to get to the center of the cell. So the key thing here is as the surface area increases, the volume increases faster. And we can see that with 3 different cell radiuses, where 1 millimeter radius, 2 millimeter radius, 3 millimeter radius, and kind of calculating the surface area to volume with that, the ratios decrease because the volume is getting bigger as you increase the surface area. So remember, volume is the amount of space something takes up, A small object has a higher ratio, which is a good thing. A larger object has a smaller ratio because as you increase the surface area, the volume increases faster. So in a ratio, if this number increases faster, then the ratio overall is going to be smaller. So when you think of a large cell, why cells aren't very big is if you have more surface area, you have more volume. And it's simply going to take too much time for something to get to the center of the cell. Nutrients, important macromolecules, oxygen, carbon dioxide, it's going to take too long to reach the center. The cell's going to die. If it is small, that means we have more surface area, less volume, and what happens then is it gets to the center faster. So it takes less time, and chemical reactions can occur faster, and the cell can maintain homeostasis much more efficiently. So this table is something that we're kind of looking at, and this is basically our cell size lab that we see right here, where we'll look at a chemical reaction, and this pinkish substance will essentially break down, and we'll see that gelatin be exposed. So the more the chemical reaction happens, you see that pink substance break down. Well, as you can see here, there is a much greater volume in the bigger cube in comparison to the surface area, where the smaller one, all that pink is pretty much completely reacted with. So it's telling us that it takes less time to reach the center of the cell. So kind of do a little bit of review. So when a cell is small, we have a large ratio. You want a large ratio. That is good. That's a good thing. So a large ratio as 6:1 is greater than 3:1, which is greater than 2:1. So this is going to show that 6:1 is the smallest cell, and a 2:1 ratio is the largest cell. Because remember, as you increase the surface area, the volume increases faster. That's what we kind of show right there. So we want to maintain homeostasis. Small cells are a lot more efficient. So if this was a Goldilocks of the three bears, the middle one is not the best. This smallest one is the best. So kind of where do we see this surface area to volume ratio? Well, a long, thin shape, there's much more surface area. In a neuron, not much volume. So neurons need to, really conduct electrical impulses very quickly, keep that homeostasis. So it is shaped specifically for that function. The folds in the intestines, once again, more surface area. Took a look at this in Unit 1. Less volume. Things are absorbing faster. Structure determines function. It is folded to increase surface area, less volume. Plant cells are also much larger than animal cells, and they increase the surface area to volume with having the central vacuole take up majority of the space. It pushes the organelles to the edges so things can get to those organelles faster. So even the plant cell as a whole is also built to be taking advantage of that surface area to volume ratio. And that's why plants want to be in a hypotonic solution. It leads to greater diffusion rates, and it leads to better efficiency, and homeostasis is maintained easier. So final thing, a little foreshadow, a little extension question. If cells do get too big, what happens? Well, if they get too big, it's no longer efficient. So what ends up happening is we have cell division. And we will talk about that in a later unit. But as cells get too big, that triggers the idea it is time to divide because it's no longer efficient at what it needs to do. So one cell becomes 2. They are small again, and as they get bigger, they'll then divide, and the process happens all over again, which is called the cell cycle. Okay, that is it for active transport and cell size. If you have any questions, please ask. Thanks for watching.